Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I will be explaining you heme biosynthesis and the regulation of heme biosynthesis. Now let's get into the details. Heme is a metalloporphyrin. So the porphyrin molecule, it contains 4 pyrrole rings as it is shown here. So each pyrrole ring, it will be having a side chain. 2 side chains will be there in each pyrrole ring. So as you see here, so there are 4 pyrrole rings that are present in this porphyrin ring. This entire ring is referred as a porphyrin ring, and individual pyrrole rings are attached to the porphyrin ring by the methanol bridges here. And in the and also as you see here, each pyrrole ring has got a nitrogen which is towards the center of the porphyrin ring, and this nitrogen it will be interacted with the ferrous iron in the heme of hemoglobin molecule. And note that the porphyrins are colored compounds, and porphyrinogens, which are oxidized forms of porphyrins, are colorless molecules. And most abundant porphyrin is the iron porphyrin, that is, and that's what I was saying. So the iron will be there in the center of this porphyrin ring, making a iron porphyrin ring, and that is what is a heme molecule. So heme is nothing but it is a porphyrin with a ferrous iron in the center of a porphyrin molecule. And one more thing that I would like to bring to your attention here is, so each pyrrole ring as say number 1, pyrrole ring number 1, 2, 3 and 4, it will have 2 side chains like as it is written here. Say number 1, that side chain number 1, side chain number 2, and then the pyrrole ring 2 like number 3 side chain, number 4, and number 5, number 6, number 7 and number 8. So 7 and 8. Number 8. So 8 side chains will be present. So that means each pyrrole ring will have 2 side chains attached. So depending on the type of side chains, they will have different types of porphyrin molecules which will come up in the heme biosynthesis process. Let's get into that. So overall, this is how the heme structure looks like. So it has got 4 pyrrole rings, number 1, number 2, number 3 and number 4, 4 pyrrole rings, and in the center you can see there is a ferrous iron, and this ferrous iron makes a coordinated bond with the nitrogens present in each of the pyrrole rings. So 4 nitrogen from 4 pyrrole ring interacting with the ferrous iron in the right in the center there. And also, as I said in the previous figure, that each pyrrole ring is attached with side chains. So there is, in this particular figure, there is a methyl vinyl side chain, then again methyl vinyl side chain, then the methyl propionate side chain and propionate methyl side chain. Look at the arrangement of side chains. So it is going in this order: methyl vinyl, vinyl is 2 carbon, then methyl vinyl, methyl vinyl. So look at the arrangement: methyl vinyl, methyl vinyl. Look at the symmetry: methyl vinyl, methyl vinyl, and then methyl, first comes methyl, then the propionate, 3 carbon molecule propionate, and then look at the 4th ring. There is some change in the 4th ring, that is propionate and methyl. So the order of arrangement of these side chains goes as methyl vinyl in the first chain, methyl vinyl second chain, methyl propionate and propionate methyl. So there is a flip in the side chain in number 4 pyrrole ring. So number 1 pyrrole ring methyl vinyl, number 2 pyrrole ring methyl vinyl, number 3 methyl propionate, number 4 there is a flip, that is propionate methyl. This is how the arrangement of side chain goes on in the heme molecule. Let's look at that, how exactly that happens. Before that, we have a table here which will give examples of heme containing proteins. We have hemoglobin, which is involved in transport of oxygen in the blood, myoglobin, which is involved in storage of oxygen in the blood, cytochrome C. Cytochrome C is part of electron transport chain, especially 4th complex in the electron transport chain. Cytochrome P450s, these are detoxification enzymes present in endoplasmic reticulums, particularly smooth endoplasmic reticulum. Then we have catalase enzyme, which is involved in degradation of hydrogen peroxide, and it is present at high concentration in the peroxisome. And then tryptophan pyrrolase enzyme, which is involved in oxidation of tryptophan. So all these are the heme containing enzymes and proteins. Now coming to types of porphyrins. Now there are different types of porphyrin intermediates which will come in the heme synthesis. So those are written here. So let's go into uroporphyrin 1, uroporphyrin 3, coproporphyrin 1, coproporphyrin 3 and protoporphyrin 3 going into heme molecule. So basically protoporphyrin 9 will go into heme molecule in fact. Now uroporphyrin, look at the side chains. Only the difference in these molecules are the side chains. Each of the 4 chiral rings in a porphyrins are attached with the 2 side chains. As I already told you, there are 3 most important porphyrins in humans, that is uroporphyrin, coproporphyrin and protoporphyrin. These are the 3 porphyrins. That's what we are going to see: uroporphyrin, coproporphyrin and protoporphyrin. Now these porphyrins, they differ in the side chain. So this is what you need to have an idea, so that as we go into biosynthesis, in biosynthesis you will see these names coming up. That's why it is important for you to know them. So in uroporphyrin, there is acetate and propionate in the first chiral ring, then acetate propionate in the second chiral ring, acetate propionate in the third chiral ring and acetate propionate in the fourth chiral ring. So that means uroporphyrin, they simply contains only acetate and propionate side chains. Acetate is a 2-carbon molecule. Propionate is a propionate. So propionate is a 3-carbon molecule. So acetate and propionate. So now look at uroporphyrin 1 converting into uroporphyrin 3, what is the change there? So when uroporphyrin 1 is converted to uroporphyrin 3, carefully look at chiral ring 1, 2, 3, 4, what's going on there? Look at chiral ring 1, acetate propionate, same. Then 2, acetate propionate, it is same. Third, acetate propionate, it is same. Look at the fourth one. In the fourth ring, there is a flip. Instead of acetate propionate, there is a propionate acetate. That's a change. That's the only change between uroporphyrin 1 and uroporphyrin 3. Remember that. Let's look at coproporphyrin. Coproporphyrin compared to uroporphyrin, look at the side chain. Instead of acetate, you are going to find methyl in all of them. So acetate is converted to methyl. So you will have methyl propionate. Methyl is 1 carbon. Methyl is single carbon. Okay. So basically there will be decarboxylation process going on in this process. So all acetates are all converted into methyl now. Now let's look at proto. And also similarly, before I go to proto, coproporphyrin 1 is converted to coproporphyrin 3, and the difference you see, it is only flip in the fourth ring. Instead of methyl propionate, you see propionate methyl. That's a flip you are going to see there. And then look at protoporphyrin. So here number 1 for chiral ring, we have methyl vinyl. Instead of propionate, you have vinyl. So methyl vinyl, first ring. Second ring also, instead of methyl propionate in the copro, you have methyl vinyl. So methyl vinyl. And then the third ring, we have methyl propionate. Same thing written. And fourth ring, propionate methyl. Okay. So there will be, while you go from coproporphyrin to protoporphyrin, again there is a decarboxylation process. Propionate 3 carbon is converted to vinyl. Vinyl is a 2 carbon. So this is how a protoporphyrin is made, and this protoporphyrin, it will be attached with the ferrous iron to make a heme molecule. So methyl vinyl, methyl propionate, propionate methyl with the ferrous iron in the center. That's a heme molecule. So this is how heme synthesis is done. Now let's move on to see the reactions proper. So heme biosynthesis occurs in almost all cells. because heme is necessary for making so many heme containing proteins that we have named in the previous slides. And major site of heme biosynthesis, as expected, it will be going on in the bone marrow, because that's where RBCs are made. So 85% of the heme synthesis will be going on in the bone marrow. The rest of the major percentage can be in the hepatocytes because of cytochrome P450s which are present in the hepatocytes. And other cells can also participate in heme biosynthesis. Any cell that has got electron transport chain, so that cell needs heme, because cytochromes present in the electron transport chain also are heme containing proteins. Now, heme is synthesized by simple precursors, that is glycine and succinyl-CoA. So these two are combined to make a heme molecule, which is a complex structure. So there are 8 reactions in heme biosynthesis pathway, 4 of them in the cytoplasm, 4 in the mitochondrial matrix. So all of the reactions in heme synthesis, they are irreversible reaction. Unlike other metabolic pathways, some of the reactions are only irreversible, all others are reversible reaction, whereas here all 8 of them are irreversible reaction. Remember them. Let's get into the very first reaction. Very first reaction in the heme synthesis, it is condensation of succinyl-CoA, as it is shown here in the figure. You know, succinyl-CoA is a TCA cycle intermediate, remember that. So whenever heme synthesis is going on, whether it is in the liver or in the bone marrow, so succinyl-CoA is taken out of the TCA cycle. So there will be a gap there in the TCA cycle. So the gap can be filled by some other reactions which are referred as anaplerotic reaction. That concept we have studied in the TCA cycle. You may refer back to that particular lecture to know what is the meaning of anaplerotic reaction. So succinyl-CoA taken out of the TCA cycle to make heme. That means whenever heme synthesis is increased, it may be because of intake of certain drugs like barbiturates, which will be detoxified in the liver by cytochrome P450s, which are heme containing proteins. So if those enzymes need to be elevated, need to be synthesized in the liver, heme has to be synthesized. So if the heme has to be synthesized, succinyl-CoA has to be pulled out of TCA cycle to provide substrate for heme biosynthesis. So TCA cycle there will be gap, and that has to be filled by gap filling reactions or anaplerotic reaction. That's how the connection goes with the heme synthesis, barbiturates and TCA cycle. Anyway, succinyl-CoA combines with glycine to make a molecule called 𝛿-amino-lavulinic acid. So there is a decarboxylation process there where the carboxyl group of glycine is removed, and the reaction, it is catalyzed by pyridoxal phosphate dependent 𝛿-amino-lavulinate synthase, which is written as 𝛿-ALA synthase. ALA means amino-lavulinate is simply written as ALA. PLP is the requirement for the reaction, and the PLP is an active form of vitamin B6. So ALA, 𝛿-amino-lavulinate synthase, is a pyridoxine dependent enzyme. So that's what I have written here in this slide. So mitochondrial PLP dependent 𝛿-amino-lavulinate synthase is the enzyme. Succinyl-CoA condensing with glycine to make 𝛿-amino-lavulinate. So ALA synthase, that is ALA-S, there are 2 types, ALA synthase 1 and ALA synthase 2. Now ALA synthase 2 is present in erythroid cells of the bone marrow, and the gene for that ALA synthase 2, it is located on the X chromosome, whereas all other tissues, they will express ALA synthase 1. So that's one of the difference that you must remember. Only erythroid cells in the bone marrow express ALA synthase 2 enzyme, and the gene for ALA synthase 2 is located on X chromosome. So remember, if there is any issue with that X chromosome gene, so it will lead to decreased activity of ALA synthase 2, and that will give rise to a disease called X-linked sideroblastic anemia. X-linked sideroblastic anemia is because of mutation in gene that is coding for 𝐒𝐵 synthase 2 enzyme. So all other tissues, including the hepatocyte, will express 𝐒𝐵 synthase 1. So 𝐒𝐵 synthase is the rate limiting enzyme and also it is the most regulated enzyme in heme biosynthesis. That's another point you must remember there. And a little more onto this 𝐒𝐵 synthase enzymes. So the loss of function mutation in 𝐒𝐵 synthase 2, as I explained to you earlier, leads to X-linked sideroblastic anemia. So what is the meaning of sideroblastic anemia? Siderosis means, sidero means accumulation of iron. Accumulation of iron in the Fe3+ form is called siderosis. Sideroblastic, red blood cells are in blastic stage and it simply accumulates iron because heme is not there to incorporate iron. So that porphyrin ring is not synthesized, so iron is simply waiting there, it kind of builds up and hemoglobin levels will be less in the red blood cell, and that type of condition we call it as X-linked sideroblastic anemia. Now, 𝐒𝐵 synthase 2 enzyme is controlled by the availability of intracellular iron. That means erythropoiesis means RBC formation. RBC formation in the bone marrow is called as erythropoiesis. So this erythropoiesis, it is dependent on the availability of intracellular iron. Because 𝐒𝐵 synthase 2 gene is controlled by intracellular iron. That's why iron deficiency, if there is a decrease in the iron deficiency, it will lead to anemia. And the most common cause of anemia across the world is iron deficiency anemia. Why? Because iron is the one that controls or regulates 𝐒𝐵 synthase 2 enzyme gene over X chromosome. So the expression of 𝐒𝐵 synthase 2 and the amount of 𝐒𝐵 synthase 2 enzyme is dependent on the intracellular iron. And note that 𝐒𝐵 synthase 2 is not feedback inhibited by end product heme. Heme do not feedback inhibit 𝐒𝐵 synthase 2. That's another important point you must remember. First point is 𝐒𝐵 synthase 2 is regulated by availability of intracellular iron at the gene level. Second is it is not feedback inhibited by heme, that is the end product of heme biosynthesis pathway. Now coming to the 𝐒𝐵 synthase 1. Unlike 𝐒𝐵 synthase 2, previous slide. Unlike 𝐒𝐵 synthase 2, 𝐒𝐵 synthase 1 is inhibited by the end product, that is heme. That's the major difference between 𝐒𝐵 synthase 2 and 𝐒𝐵 synthase 1. Now, hemine, which is an oxidized product of heme, it decreases the activity of hepatic 𝐒𝐵 synthase 1. So that means like feedback inhibition done by heme, end product heme on 𝐒𝐵 synthase 1, the oxidized product of heme called as hemine also have a feedback negative effect on 𝐒𝐵 synthase 1, especially for hepatic 𝐒𝐵 synthase 1. So that's why hemine can be used as a treatment in some porphyrias, which are inherited conditions of defect in heme biosynthesis. The inherited disorders of defect in heme biosynthesis are called as porphyria, and certain type of porphyrias can be treated by giving hemine, because hemine has got a feedback effect, negative feedback effect on hepatic 𝐒𝐵 synthase 1. And also drugs like barbiturates significantly increase the expression of 𝐒𝐵 synthase 1, because barbiturates will be detoxification of barbiturates. It will be done by cytochrome P450 enzymes, and these cytochrome P450 enzymes are heme containing enzymes, and that heme has to come from 𝐒𝐵 synthase 1, very first enzyme in the heme synthesis, that's in the hepatocyte. And for barbiturates to undergo detoxification, so they should have sufficient cytochrome P450. For that, cell should synthesize sufficient heme, and that should come from 𝐒𝐵 synthase 1, which is the most regulated enzyme in the heme synthesis in the hepatocyte. So That's why barbiturates will induce ALA synthase 1 activity and by acting at the gene level. Let's move on to see the second reaction. So once we get ALA, that is delta amino levulinic acid, two delta amino levulinic acids, they condense with one another. This is a condensation process where there will be dehydration, means water molecule is removed, while two delta amino levulinates are condensed, as you are seeing here. Two delta amino levulinates are condensed. So this is one side, one delta amino levulinate, on the other side another delta amino levulinate. Both are condensing now. So to make a molecule called porphobilinogen. So this is the first pyrrole ring that is synthesized, porphobilinogen. So this is the dehydration process done by delta amino levulinate dehydrogenase. So with this dehydration, two delta amino levulinates are condensed to make a pyrrole ring, which is a porphobilinogen. And note that this porphobilinogen, so it has got 2 carbon molecule attached and then 3 carbon molecule attached. So these 2 and 3 carbon side chains can be further modified later. Alright, so because we have seen in this process, as you see here, acetate propionate, acetate propionate, acetate propionate, acetate propionate in the uroporphyrin 1. Look at the structure here. Look at these side chains. So 2 carbon acetate and 3 carbon propionate. So we have that acetate and we have the propionate now. So we are going towards that complex structure that I have shown you previously, this one. Look at this complex structure. So ultimately our intention is to make a very complex structure, that is a porphyrin ring with the ferrous iron in the center, that is heme, right? So the side chains we are going towards is methyl vinyl, methyl vinyl, methyl propionate and propionate methyl, right? So we are starting there in that process. So we have this pyrrole ring, the porphobilinogen, which is a pyrrole ring which has got now acetate and propionate. Let's keep that in mind. So how many pyrrole rings we need to make one protoporphyrin 9 molecule to make a heme? So we need 4 pyrrole rings. So 4 pyrrole rings are necessary. So that means 4 porphyrinogens are needed to get those 4 pyrrole rings. So that's how the heme synthesis, which is going on in the 4 reactions. First 4 reactions are going on in the matrix of mitochondria. So we are condensing all these succinyl-CoA glycine to make delta amino levulinate. Both of them condensed to make porphobilinogen. So like that we have one porphobilinogen. So other heme biosynthesis pathway brings one more porphobilinogen. Third biosynthesis of heme biosynthesis pathway takes one more porphobilinogen. So multiple heme biosynthesis pathways will be going on in the mitochondrial matrix making porphobilinogen. Now 4 porphobilinogens, they need to come together to give that 4 pyrrole rings and make a uroporphyrinogen molecule. So right now we have only porphobilinogen, 1 pyrrole ring. To make uroporphyrinogen, say uroporphyrinogen, we need 4 pyrrole rings so that uroporphyrinogen is made. So for that 4 porphyrinogen has to be brought by 4 heme biosynthesis pathway up to this stage. Okay, so here is the brief review of reaction number 2. So 2 molecules of delta amino levulinate condensed to make a pyrrole ring, that is a porphobilinogen. Delta amino levulinate dehydratase is the enzyme that conducts this reaction, and delta amino levulinate dehydrogenase is also called as porphobilinogen synthase. And this delta amino levulinate dehydratase or PBG synthase, PBG means porphobilinogen, so which is the product of this reaction called porphobilinogen. That's why the name of the enzyme is porphobilinogen synthase. So delta amino levulinate dehydratase or PBG synthase, importantly, it's a zinc containing enzyme and also it is very sensitive to lead. And whenever lead binds to this enzyme, it's going to replace the zinc and lead will bind there, and thereby it will inhibit 𝛿-aminolevulinic dehydratase enzyme. That's how lead toxicity will inhibit heme synthesis. That's how lead can act as a toxic effects in our body. So lead toxicity increases 𝛿-aminolevulinic because it do not allow two molecules of 𝛿-aminolevulinic to condense with one another. Thereby, it decreases heme biosynthesis, leading to decrease in hemoglobin synthesis. Now let's see how a uroporphyrinogen molecule is made. So as I said before, 4 molecules of porphobilinogens, so they will condense with one another. So we are bringing 4 molecules of porphobilinogen now, as I was explaining before. 4 molecules of porphobilinogen will come from 4 heme biosynthesis reaction, and they will undergo condensation where the ammonium ion will be released, that side chain NH2 will be released as ammonium ion, and we get hydroxymethylbilane, right? 4 porphobilinogens are arranged one besides the other, and we make a linear molecule called hydroxymethylbilane, which is a linear tetrapyral molecule. So this particular reaction is catalyzed by uroporphyrinogen 1 synthase, and this hydroxymethylbilane will undergo cyclization process, which is catalyzed by uroporphyrinogen 3 synthase to make a type 3 uroporphyrinogen. So the cyclization process of linear tetrapyral is 4, it will be like this. So all of them are connected, side chains are there, and this is what is hydroxymethylbilane, HMB. So this is done by uroporphyrinogen 1 synthase, which is also called as HMB synthase, because HMB is synthesized. Alright, so HMB synthase making hydroxymethylbilane, so where it's a linear tetrapyral as I have written here. So this cyclization has to go on, that linear tetrapyral has to be cyclized, means ring has to close, and that will be done by uroporphyrinogen 3 synthase to make type 3 uroporphyrinogen. It can also happen by a spontaneous process. Spontaneous cyclization can also happen, and that will produce uroporphyrinogen 1. So type 1 uroporphyrinogen is by spontaneous process. In our cells, predominantly the uroporphyrinogen 3 synthase is the one that will cyclize hydroxymethylbilane, and you get type 3 uroporphyrinogen molecule. Now this type 3 uroporphyrinogen, as you see, it contains acetate propionate, and then there is a propionate and acetate molecule. So that's the type 3 uroporphyrinogen, which is converted into uroporphyrin, type 3 uroporphyrin by flipping up the ring there. Now in the reaction number 3, we have 4 porphyrinogen condenses with one another to make a linear chain containing acetate propionate molecules. So which is done by porphobilinogen deaminase, because there is a deamination process, which is also referred as HMB synthase, and also in the previous figure it is also sometimes referred as uroporphyrinogen 1 synthase. These are the same names here, for 3 names for same enzyme, because we have deaminated porphobilinogen, because as you see here in the reaction, porphobilinogen is undergoing deamination. There is a deamination here. That's why this enzyme can also be called as porphobilinogen deaminase. And also there is a synthesis of hydroxymethylbilane, HMB is synthesized, right? So it is called HMB synthase. And since uroporphyrinogen 1 can be made from it spontaneously, that's why this enzyme can also be referred as uroporphyrinogen 1 synthase. Okay, so same reaction, 3 enzyme names here. Same enzyme referred in 3 ways. Remember that, because in the exam some examiners might change these names. So porphobilinogen deaminase, HMB synthase and uroporphyrinogen 1 synthase, it is all same. Alright. So now let's move on to number 4 and number 5 reaction. Number 4 reaction is uroporphyrinogen 3 co-synthase catalyzes the flipping of A and P groups at the 4th chiral ring. So that's what is seen here. As you see, I am taking you back to this slide. Look at this. Number 4, 4th ring, acetate, instead of acetate propionate, there is a propionate and acetate. So, because acetate propionate, acetate propionate, acetate propionate and propionate acetate. So, that flip is going on, and that flip, it will be done by uroporphyrinogen 3 co-synthase enzyme. That is the one which causes the flip in the ring. Okay, and that flip is shown here in uroporphyrinogen 3. As you see, the 4th ring, there is a propionate and acetate. Okay, now that's number 4 reaction. Number 5 reaction is uroporphyrinogen decarboxylase enzyme catalyzes the decarboxylation of all the acetates into methyl groups. Acetate is a 2-carbon molecule, methyl is a single carbon. So there is a decarboxylation of all acetates. How many acetates we have? We have 4 acetates. As you see, in each chiral ring there is one acetate. All of them will undergo decarboxylation into methyl in each one of them. That means 4 carbon dioxide molecules will go out. And this is catalyzed by uroporphyrinogen decarboxylase enzyme. That's how uroporphyrinogen is now converted into coproporphyrinogen 3. So uroporphyrinogen 3 into coproporphyrinogen 3 is done by uroporphyrinogen decarboxylase. Make sure you remember the name of the enzyme. That's how uroporphyrinogen 3 goes into coproporphyrinogen 3. Now, coming to reaction number 6 and 7. In the reaction number 6, there is a coproporphyrinogen oxidase enzyme. In the mitochondria, it catalyzes decarboxylation followed by oxidation of a propionate in the 1st and 2nd ring to vinyl group. That makes a protoporphyrinogen 9 molecule. So because, let me take you back to this slide here. As you see, conversion of coproporphyrinogen 3 into protoporphyrin 3 molecule. Look at this. Some of the rings that are present. So there is methyl propionate, and propionate methyl. So there is propionate number 1, number 2, number 3, number 4 rings have got propionate, which is 3 carbon molecule, right? So look at protoporphyrin 3 molecule here. So number 1 ring has, instead of propionate, there is a vinyl. Number 2 ring has, instead of propionate, there is a vinyl here. So that is what is going on in number 6 reaction, where number 1 chiral and number 2 chiral rings which have got propionates, and propionates will be converted to vinyl groups, because propionate is a 3 carbon, vinyl is 2 carbon, so one of the carbon is removed as carbon dioxide. So 3 carbon to 2 carbon molecule. That's what happens with coproporphyrinogen oxidase enzyme. Number 7 reaction is oxidation of protoporphyrinogen 9 into protoporphyrin 9 catalyzed by protoporphyrinogen oxidase. So that's number 7 reaction. Let's move on to number 8 reaction. So we have a protoporphyrin 9. This protoporphyrin 9 will be incorporated with an oxygen ferrous-iron retic. This particular protoporphyrin 9 is incorporated with the ferrous iron, that is done by ferrochelatase enzyme, and that's how the ferrous iron is there in the center of molecule, and this is how we have made heme. So protoporphyrin 9 is converted to heme by ferrochelatase enzyme, that's given here, and so protoporphyrin 9 is converted to heme by ferrochelatase enzyme by incorporating ferrous iron into the center of protoporphyrin 9. So this is what is the heme molecule. Now heme has got methyl, vinyl, methyl vinyl, methyl propionate and propionate methyl side chains. Alright, so that's how the heme is made. So incorporation of ferrous into the protoporphyrin catalyzed by mitochondrial ferrochylase enzyme. Note that this ferrochylase is a lead sensitive enzyme. So lead is going to inhibit this enzyme. So protoporphyrin 9 will bind with the zinc that is released by delta amino dehydratase by lead to form zinc protoporphyrin, which is found to be elevated in lead poisoning. So whenever there is a lead toxicity, so you remember the second reaction, ALA dehydratase enzyme, which is lead sensitive, and ALA dehydratase is a zinc containing enzyme. So lead will bind to ALA dehydratase and release the zinc, and that zinc will come and bind to protoporphyrin 9 because same lead, it is inhibiting ferrochylase enzyme. In the last reaction, do not allow ferrous iron to bind to the center of the protoporphyrin 9 molecule. Instead, zinc will come and bind there. Okay, so here in the presence of lead, okay, let me show you that. So in the presence of lead, instead of ferrous iron, zinc will bind in the center of this protoporphyrin 9 and that will become zinc protoporphyrin 9 molecules. So this zinc protoporphyrin 9 will be elevated in lead toxicity along with delta ALA aminolevulinate. So there are two things that are elevated in lead toxicity. One is delta ALA, other is zinc protoporphyrin 9, because lead is going to have an inhibitory effect on ALA dehydratase and ferrochylase. So here is the overview of heme biosynthesis. So 8 reactions, 4 of them are going on in the matrix of mitochondria, 4 of them are going on in the cytoplasm. Okay, first reaction and last 3 reactions going on in the mitochondrial matrix. So the first reaction is catalyzed by delta aminolevulinate synthase, condensation of succinyl-CoA with glycine. Remember, succinyl-CoA come from TCA cycle. Let me write that too also here. You know, TCA cycle is running in the mitochondrial matrix itself, so it takes it from there. So delta aminolevulinic acid is going out of the mitochondria into the cytoplasm. So in the cytoplasm, delta aminolevulinate is going to condense with one another to make porphobilinogen done by delta aminolevulinate dehydratase. It's a zinc containing enzyme and it is sensitive to lead. Porphobilinogen, 4 of them will condense with one another and release ammonium ion done by porphobilinogen deaminase, which is also called as HMB synthase because HMB is synthesized, and also it is called as uroporphyrinogen 1 synthase. So HMB is hydroxymethylbilane, which is a linear tetrapyrrole, and it will undergo cyclization process. So to make uroporphyrinogen 3. Enzyme is uroporphyrinogen 3 co-synthase. Now the uroporphyrinogen 3 undergoes decarboxylation process and converting all their acetates into methyl groups. So we get coproporphyrinogen 3. Coproporphyrinogen 3 enters into the matrix of mitochondria and it will undergo oxidation and decarboxylation to make protoporphyrinogen 9 molecule, and protoporphyrinogen 9 undergoes further oxidation to become protoporphyrin 9, and finally protoporphyrin 9 will get ferrous iron incorporated by ferrochylase enzyme making heme, and ferrochylase is also lead sensitive enzyme. So this is how heme synthesis is done. Now let me explain you something called Soret band. Porphyrinogens are colorless molecules while their oxidized products called porphyrins are red in color. Remember this. This will help in the diagnosis of porphyrias, inherited conditions of heme biosynthesis called porphyrias. In the diagnosis of porphyrias, this red colored molecules appearance in the urine will help in the diagnosis. Porphyrinogens are colorless, porphyrins are colored molecules. The visible and ultraviolet spectra of porphyrins and porphyrin derivatives are useful for their identification. So the sharp absorption band near 400 nanometer a distinguishing feature shared by all porphyrins. It is termed as Soret band. That means at 400 meter, that means at 400 nanometer range, there will be maximum absorption of light by porphyrins, which is something not done by porphyrinogens. And porphyrins, with this characteristic absorption at 400 nanometer range, will help in the identification and determination of porphyrin levels in the solution. And that band which is shown here is referred as Soret band. Remember that. Alright, now let me explain you little more into regulation of heme biosynthesis, although we have covered this topic in the very first reaction itself. Synthase is the key regulatory enzyme in heme synthesis, and end product heme regulates only synthase 1. Heme do not have any effect on synthase 2. Synthase 2 is regulated by intracellular iron. I have already explained that to you. Heme and hemine. Heme is ferrous iron in the center, hemine is ferric iron in the center. That means oxidized product of heme is hemine. So both heme and hemine, it inhibits activity of pre-existing synthase 1 by feedback negative effect. Heme also diminishes the transport of synthase 1 when it is synthesized in the cytoplasm, the new enzyme synthase 1, which is made in the translation going on in the cytoplasm. Then it has to go into the mitochondria to participate in heme biosynthesis. So having heme in the cytoplasm, it will have a negative effect on synthase transport into the matrix of mitochondria. Heme also represses production of synthase 1 at the gene level, and synthase 1 enzyme synthesis is upregulated by large number of drugs, example barbiturates, because I told you barbiturates, they are going to be detoxified by cytochrome P450 enzymes, and cytochrome P450s are heme containing enzymes, especially in the hepatocytes. So barbiturates in the hepatocytes will induce synthase 1, thereby they will induce CYP450 enzymes. Okay, that's how they will be undergoing detoxification. So this is how some of the drugs like barbiturates, they upregulate synthase 1 activity. And also vitamin B6 availability will control synthase 1 activity. And more importantly, the last but not the least is activity of synthase 1 is increased by fasting and starvation, whereas feeding of carbohydrates will decrease the activity of synthase 1. So meaning whenever person goes into the fasting condition and starvation, so there is more increase in synthase 1. Why? Because there is a need for heme synthesis during fasting and starvation. So more heme synthesis enzymes like cytochrome P450s, electron transport chain cytochromes, all these things need to be increased. So that means heme synthesis has to be increased, and that means more and more heme synthesis has to go on. So synthase 1 will be activated. So the glucose feeding will decrease the activity of synthase 1 because it's going to repress synthase 1 gene. So fasting and starvation increases synthase 1 activity, whereas feeding of carbohydrates, especially like if a glucose infusion is going to decrease synthase 1 activity by repressing the gene that is coding for synthase 1. So that's another point that you need to remember. Now coming to synthase 2. Synthase 2 enzyme is in the erythroid cells and is not regulated the way synthase 1 is regulated. In erythroid cells, heme synthesis is one-time event and it stops when red blood cells mature. So heme also stimulates protein synthesis, including synthesis of enzymes of heme biosynthesis. So thereby heme combines with globin to make hemoglobin, and this will ensure the equal quantities of heme and globin is made to assemble hemoglobin protein. So this is all about all the points, regulation, reactions, everything about heme biosynthesis and regulation of heme biosynthesis. I hope this lecture has helped you in understanding this very important topic on heme synthesis, because this particular topic is connected with so many other clinical applications, especially porphyrias, that we are going to see in the next lecture, and also the way barbiturates regulate heme synthesis, how fasting and starvation affects our heme biosynthesis process, especially in the hepatocytes, how glucose infusion is affecting, how heme and hemin is having an effect on allosynthetase 1. All of them will have a clinical application. Thank you.